Alright, welcome back to our third and final installment on enzymes. So today we're just going to focus on regulation. We've covered the basics, and it's time to kind of go down the rabbit hole of how we biochemically tune these enzymes to get them to perform reactions when we want them and at rates at which are biologically advantageous, and to coordinate them with all the other functions that are intertwined metabolically, which is quite the feat. So some of the things we're going to talk about today is what factors influence enzymatic activity. We're going to spend quite a bit of time with allosteric regulation. So what are the general features of allosteric regulation? What makes it unique from other types of regulation, how they can work together. We are going to go through two models of allosteric regulation, which have different explanatory strengths and weaknesses, and I'll give you my opinion on that. We'll talk about covalent modification, which is a really important type of regulation for enzymes. We'll talk about how both covalent modification and allosteric regulation work in concert, with a couple classic examples, and then we're going to spend a little extra focus time on hemoglobin, because it's hemoglobin and myoglobin are really the classic example of how allosteric regulation functions. And there's nearly always a hemoglobin question on any standard biochemistry exam having to do with allosteric regulation. So we're going to spend some time there because I think it's part of the biochemical canon, so to speak. So what are some of the factors which influence enzymatic activity that we should just be aware of before we start to go into more detail? Well, first and foremost, availability of substrate is incredibly important. If you don't have substrate and you don't have cofactors that are required, then the reaction can't proceed. But if you have an abundance of substrate and you have the required cofactors, that will not be a limit to the speed of the reaction. Also, the product matters. As product accumulates, it's very common for the rate of an enzymatic reaction to decrease because of feedback regulation, in which the product actually acts as a regulator and turns down the rate of reaction so that you don't overaccumulate product. It's basically just an off switch when you have enough of what you need. And this is really important for energetic efficiency. There's the mechanism of genetic regulation, which includes synthesis of new enzymes and decay of enzymes. And this is very energetically expensive and relatively slow. So in terms of long-term, half-day long, full-day long, 3-day long adaptations, this makes a lot of sense to acclimating an organism to a particular environmental need. But in terms of immediate response, it's quite slow. And so we lean on the ability to regulate via allosteric and covalent mechanisms much more commonly for fast adjustments. And having said that, there's allosteric regulation, there's regulation through covalent modifications, and there's also a whole host of zymogens, isozymes, and other proteins which modulate effects of proteins specifically as their main biological function. So one of the factors that is kind of unique, so I'm just going to put it on its slide right up front, and then we're going to walk away from it for the rest of the lecture, is the idea of isozymes having different subunits that give enzymes unique specificity to different tissues. The really good example here is lactate dehydrogenase, which has 4 subunits, and the subunit can be either subunit A or subunit B in any combination. So if you have an A4 isozyme of lactate dehydrogenase, that means that all 4 subunits of the LDH is the subunit A. Conversely, if you have 4 identical subunits of subunit B, that would be the B4 isozyme, and then you have every combination in between. Now, why this is important is because the function of lactate dehydrogenase is to convert lactate to pyruvate as part of anaerobic metabolism. And so it's ubiquitous in all tissues. And almost all of our tissues see some amount of lactate dehydrogenase expressed. Some substantially more than others, obviously. But the proclivity for it to do its job isn't identical between all tissues. So there's little tuning that's happening with having different subunits which affect its function in its environment, and it can fine-tune that function based on the ratio of A to B subunits. For instance, liver, let me grab a pen here. Liver is A4, so is muscle, and it has a higher expression than muscle. And then if we look at heart, heart is B4 and has a higher expression than kidneys, even though kidney has very high LDH. So the function of LDH and the quantity expressed is variable and tissue dependent. Another neat thing about this chart is it's not exclusively A4 in liver. You can see that we actually have a little bit of A3B and a little bit of A2B2. Conversely, if we go and take a look at heart, not only do we have a vast majority of B4, but we also have a pretty reasonable amount of AB3 and a little bit of A2B2. So you can fine-tune the tissue's function not only by changing the ratio of isozyme, or the isozyme itself, but the ratio of isozymes expressed in the tissue. So this is a really neat modulatory control for some of these ubiquitous enzymes. So let's just jump right into the meat of it and talk about allosteric regulation right out the gate. An allosteric regulation is fundamentally just an action at the enzyme on a different site, other than the binding site or active site of the enzyme. And so it's very common for enzymes which are situated in metabolic pathways where you have sequences of reactions taking place to be modulated by allosteric effectors. And these effectors come from other points in the pathway, either preceding it or following it. And so how this pathway is moving from step to step to step, accumulating substrate and accumulating product, will affect the way the enzyme functions. And this elegantly affects how the enzyme consumes those substrates to produce those products. So, and these effectors can be either forward activators or they can be feedback inhibitors. So you get pressure from both sides of these sequence of events. And one of the characteristic aspects of what makes allosteric regulation unique is it has a sigmoidal kinetic to it. So this red line here is a sigmoidal curve compared to the hyperbolic curve that we've been accustomed to looking at. And this is a departure from the characteristics of classic Michaelis-Menten kinetics. So you can always identify what is kinetically allosteric effectors just by looking at the concentration of substrate versus the velocity of reaction and seeing the sigmoidal curve as indicative of allosteric regulation. So there's two models which explain allosteric regulation, and the first one is the Monod-Wyman-Chang'u model, and that's classically called the MWC model. And this is, in terms of the way you visualize protein conformational changes, perhaps not the most accurate model, but it has really good predictive power for a couple classic enzymatic systems. So I just want everyone to realize that just because something's a model doesn't mean that it's a statement of truth. Models just provide predictive power. Good models provide us excellent predictive power, which often gives us mechanistic insight. But we don't always get to mechanistic truth via a model that has predictive power. So if this sounds a little out of sorts in terms of chicken and egg rationale, It probably is, but it still doesn't mean that this doesn't have good predictive power. So, and it doesn't mean it's completely devoid of a little bit of rationale for the function either. So, under the MWC model, allosteric proteins exist in two forms. They have relaxed forms and they have taut forms. And the relaxed form is more capable of binding the enzyme and the substrate, and the taut form is less prone to binding. And so the taut form predominates in the absence of substrate. And when substrate binds, it's going to bind much more tightly to the relaxed form than the taut form. So there's a preference for the substrate in the relaxed state. Let me just move on to the next slide here. Here we go. A little bit more detail on the MWC model is, if we're getting oriented on this slide, starting top center here, we have dimeric protein, which it's going to start out in the relaxed form, and it is in equilibrium with the taut form here. And you can see a difference in the way that activators, substrates, and inhibitors all interact with these different forms. So if you're starting from a relaxed state, you have a high affinity for activator, which then increases the affinity for the substrate. Likewise, if we go, that's going down this way. If we go this direction, you have a high affinity for the substrate, which also increases the ability to bind the activator. And this combination of effects actually feeds in on itself to cause improved binding of both. Whereas if you are in the taut form, you have an improved ability to bind inhibitors, which has a feedback loop, making the taut form less accessible to substrates and activators, and slows everything down even more. So this feedback is considered a sequential, is a little bit hard to digest for me, because actually, let me just hop back to the previous step. Because the idea is that you have an equilibrium between forms, which influences how it interacts with substrates, activators, and inhibitors. And that's not actually too much of a stretch, because we know that enzymes in their preferred states are incredibly energetically similar to other states that are nearly the same energy level. So you can have a state that you could consider relaxed and tense, or R and T, and you could envision an equilibrium between these two very similar energetic states, and that equilibrium would dictate how it's binding substrates, activators, inhibitors. And as soon as you have a substrate or activator bound, it's going to give you a little bit of feedback, which is going to really commit it to one direction. And if it's binding an inhibitor, it likewise will commit it towards the opposite direction. Now, the way I've explained proteins and typically envisioned them myself is more along the lines of the sequential model. So this is the alternative model proposed by Koshland, Nemeth, and Filmer. It's the KNF model. And the idea behind the KNF model is that instead of having multiple similarly energetic levels that are in equilibrium, one that is got a higher affinity for substrate and activators, and one that has a higher affinity for inhibitors, the idea is that the interaction of the preferred state of the protein with a substrate, or with an inhibitor, or with an activator, causes a conformational change. And this is the way I typically envision these structures, because if we go back and we think about this a little bit more clearly, you know intuitively at this point that the binding of the substrate to the R conformation is going to cause a change to its structure inherently. Likewise, the binding of an activator will cause a change to structure. Just because you potentially have an equilibrium existing between R and T doesn't mean that the transitions that we're looking at in this model don't involve small or perhaps large and meaningful conformational changes as the activator, substrate, and inhibitors are binding. So perhaps like so many things, some truth can be extrapolated from one and some truth can be taken from the other, and by using both models we can get the best picture of what's going on. But I tend towards thinking of it a little bit more like the KNF model. And the reason for that is because we know when you have an association, covalent or otherwise, with a small organic compound, with a protein, that will change, ultimately change, the way it has preference to fold. Excuse me. And therefore, we'll have a new preferred lowest energy state in its tertiary conformation, and that will change its structure function. So, the idea behind this is that if you have a symmetric dimeric protein, let me grab a color here, and it binds a substrate, the binding of that substrate will change one of the dimers to a substrate-bound dimer, which is paired dimerically. Now, as that single monomer is changing to substrate-bound conformation, it's likely, and I would argue inevitably, going to translate conformational change to the other dimer or other monomer which is bound as part of the dimer. Now, in terms of cooperativity, if the relative affinities for the various conformations for S are ordered like what we see here, then we have a positive cooperativity, where we have an equal or greater than relationship between the modified and substrate bound variations that are both higher than the non-bound monomer. However, if the relative affinities for the various conformations are ordered like this, where the substrate bound dimer has greater affinity than the symmetric unbound monomer, but it has a greater affinity than the modified transmitted conformational change, then this would be considered a negative cooperativity. And this is just a basic kinetic plot, Wolff-Kirchner plot, of this phenomenon, where you have in blue a plot of no cooperativity. If you have negative cooperativity, where the binding of the substrate affects one monomer positively but downregulates the other, in purple, and then in red you see positive cooperativity. So what's the most notable differences between these models other than what I've already discussed? In the MWC model, the different conformations have different affinities for different ligands because of that equilibrium between them. And the concept of ligand-induced conformational change is ignored. However, in the KNF model, when you bind ligands, we are taking into consideration the fact that there is going to be conformational changes taking place. And even though I lean towards the KNF model in terms of explanatory power, the way I think of protein structure, function, and regulation, there is likely a lot to be said for a balance between these two, and some enzymatic systems probably lean more towards the MWC model than KNF, and some lean more strongly on the model supplied by KNF and less on the MWC. So it's always a good idea to think of both of these simultaneously and try to extract the most information we can to get the most sound conclusions. So moving on from these models of allostery, what kind of covalent modifications regulate enzymes? Because we talked about covalent modifications at the very beginning, saying these are very important. The most dominant, most important covalent modification that we should pay attention to is enzymes being regulated via reversible phosphorylation. So proteins can, depending on the enzyme and protein, be regulated through single phosphorylation events or many, many, many phosphorylation events. And the degree of phosphorylation is often like a rheostat that slowly turns up or slowly turns down a protein's degree of activity. And so this is the most common form of covalent modification, adding phosphoryl groups to the outside of the protein, thus changing its three-dimensional structure, thus altering its function, whether up or down. And there's a whole host of enzymes which are specific to performing these phosphorylation modifications on enzymes as regulator proteins. And these proteins are called kinases. So, a protein kinase. Not only are there kinases, there are kinases for kinases and kinases for kinases for kinases. And you'll see these listed as protein kinase, PK, protein kinase kinase, PKK, kinase kinase kinase, KK, etc. So, kinase, the use of the modification of proteins by kinases to phosphorylate proteins is very, very important in terms of our multiple metabolic pathways. Now, how do you dephosphorylate? Well, protein kinases put phosphates on. If you want to dephosphorylate, you use a phosphoprotein phosphatase or simply phosphatase. And this is the reverse reaction where you're removing phosphoryl groups from proteins. So, like I said before, kinases are highly regulated, phosphatases are also highly regulated, and many of them are regulated by phosphorylation events of their own. So, the details of this is actually beyond this particular class. If you take immunology, you'll get a lot of insight into how the immune system and inflammatory mechanisms are highly regulated through the kinase phosphatase pathways. So, protein kinases phosphorylate specific residues. We've got phosphorylation taking place at serines, threonines, tyrosines. Those are the three targeted by kinases, and therefore also targeted by phosphatases in reverse. Despite the specificity of these residues, not all kinases will attack all serine, threonine, or tyrosine residues. There's a lot of catalytic specificity which is unique to each target. So you have some conservation of the core mechanism, but we also have a lot of diversity in how it's applied. So in this picture below, what you see is a PKA, or protein kinase A, and it's shown complexing to this orange pseudosubstrate peptide here, which is a peptide which is acting like a substrate and occupying its binding pocket. And this kind of pseudosubstrate peptide is one of the key ways PKA and other kinases are regulated. Sometimes the body needs to provide a substrate in replacement, but it's very specific replacement peptide versus this actual target peptide. Let me see here. I think I got out of sequence a little bit. I did get out of sequence here. Let me do something real time. Okay, we're just going to skip this slide, then we'll come back. So, phosphorylation is not the only form of covalent modification. There's a variety of other common, but not as common as phosphorylation modifications that can take place covalently to change the way a protein is functioning. So these can include adenylation, uridylation, methylation, oxidation reduction, which can be your cysteine residues, and acetylation, and complexation with ADP ribosylation. Methylation, very common. Redox, very common for cysteines. Acetylation, very common. Adenylation, and the others, a little less common, but all very, very important depending on the system you're studying. We're not going to go into too much detail, just be aware of these being other modes of modifying proteins covalently to change their structure function transiently as part of our metabolic regulation of enzyme function. Now, going back up here, or perhaps here. A good example of regulation that we want to pay a little bit of attention to. Let's talk about glycogen phosphorylase as an example that we can lean on a little bit for a system that has both allosteric regulation and is subjected to covalent modification, where both of these types of regulation take place. So, what is glycogen phosphorylase? It cleaves glucose units from the non-reducing end of glycogen. So glycogen is this polymer of carbohydrates that we use and store in our muscles and liver for fast-acting blood glucose production or glucose production in the muscle during high-intensity exercise. And this converts the glycogen to a form of useful fuel, glucose 1-phosphate, which is produced here by cleaving there. And this is affected by both allosteric regulation as well as deliberate covalent modification. Now, another step here is that glucose 1-phosphate is converted to glucose 1,6-phosphate as part of subsequent metabolism. So when you see glucose 6-phosphate potentially acting as a feedback regulator, allosteric regulator of the system, that's why it's in play. I'm just going to try this one out here. Let's see, cyclic AMP. So cyclic AMP, let's see. I'm just all over the place on you guys today. Sorry for that. Okay. Well, I'm just going to go with this. Executive decision, going back. Sorry. Okay, now we're on the right spot. We're just going to go with it. Cyclic AMP is a signaling compound derived from cleaving common nucleotides and forming a non-energetic cyclic AMP compound, which acts as a secondary messenger or signaling molecule. And what it does is it is a protein regulator. All right, I thought I had everything mixed up, but apparently it was right the first time. When I was looking at this, what got me confused was that I was focused on the function of cyclic AMP in regulating the phosphorylation of the system we're discussing. And what I realized is this slide is here specifically just to introduce the notion that cyclic AMP acts to activate protein kinase A. So this slide is pertaining to cyclic AMP-dependent protein kinase, also known as pKa, which is the tetramer we were discussing. And when you have an abundance of cyclic AMP, the tetramer gets broken up because the cyclic AMP binds the regulatory subunits, releasing a pair of the catalytic subunits, which are active as monomers. So that's just background, which will make sense in a couple more slides. We talked about other modes of non-phosphorylative covalent modification. Then we started to talk a little bit about the allosteric and covalent modification of glycogen phosphorylase. Here we go. And now jumping into a little bit more of the nitty-gritty on the structure of phosphorylase, glycogen phosphorylase. So it's a dimer of identical subunits. Each subunit contains an active site, so both subunits can bind at an active site, and also have a point for allosteric effector, which is near the subunit interface. There's a regulatory phosphorylation site located at a specific serine for each subunit, and there's also a glycogen binding site, which exerts regulatory control. Now, you can see here that each of these subunits contributes a tower helix, and these tower helices extend from their respective subunits and also pack against one another. So we have all these different sites that we've been discussing with this tower helix. We've got an AMP, allosteric effector site. We have a phosphorylative site, catalytic site here. We have a glycogen binding site. So the overall gist of this is that, yes, glycogen phosphorylase is regulated allosterically. So muscle glycogen phosphorylase shows a cooperativity in substrate binding. ATP and glucose 6-phosphate, remember, which is one step away from the glucose 1-phosphate, are allosteric inhibitors of glycogen phosphorylase. So ATP, glucose 6-phosphate are allosteric inhibitors. And that metabolically makes sense because of the action of glycogen phosphorylase is to cleave units of glucose 1-phosphate so that we have this newly liberated source of energy. Once we have a surplus of energy, we have a good biological reserve of ATP. We have a good biological reserve of the metabolite one step away from glucose 1,6-phosphate as glucose 6-phosphate. This reserve acts as an allosteric inhibitor, binding to the glycogen phosphorylase enzyme and reducing its rate of reaction. Conversely, AMP is an allosteric activator of glycogen phosphorylase. And this makes sense because AMP indicates that not only do we not have a lot of ATP, we're also perhaps reduced in ADP because we've reduced the degree of phosphorylation of these compounds to such a dangerous extent that our energy status cellularly has been affected. So it's incredibly important that in the presence of AMP, glycogen phosphorylase activation increases substantially to liberate as much glucose 1-phosphate from glycogen as possible. Now what's interesting is that in terms of the two models which we discussed initially, glycogen phosphorylase conforms more closely to the MWC model than the KNF model. And that means that we have this control, covalent control, which in this case isn't just an equilibrium, but we have a covalent control which designates the T state and the R state. And this is just a really nice diagram because you can see how the phosphorylation of glycogen phosphorylase leads to one type of regulation, and then the interaction with all of these other modulators, whether it's the glucose 6-phosphate, the glucose itself, or caffeine, can affect the way that the phosphorylated or non-phosphorylated versions react. So you have this combination of allosteric control as well as covalent modification. Now, how does this covalent modification occur? Well, that's why I wanted to introduce the concept of cyclic AMP a little bit earlier. What happens is the phosphorylation is mediated by a cascade of enzymes. And what we're focused on here is this conversion of an inactive phosphorylase kinase becoming active, and this takes the inactive glycogen phosphorylase and converts it to the active glycogen phosphorylase. But preceding it, you can have any kind of hormone signaling take place, and it'll activate a hormone called Adenylyl cyclase, which will actively convert ATP to cyclic AMP directly, not through consuming it for energy, but by modifying it structurally. And the cyclic AMP then activates the cyclic AMP-dependent protein kinase, or pKa. The pKa then causes a phosphorylation event on the phosphorylase kinase, and this allows for the activation or phosphorylation of our glycogen phosphorylase. So like I said, there's a series of reactions that take place that amplify quickly. And this is just the covalent modification. Please don't commit this to memory, but know that when you have ATP and it interacts with adenylyl cyclase, that you have a simple enzymatic attack where a base pulls a proton, transfers the electrons to the oxygen, which then subsequently cause a dephosphorylation reaction and a cyclic product, releasing pyrophosphate, which is a pair of covalently linked phosphates. So let's change over now to a different protein, hemoglobin. And the reason we're going to focus on hemoglobin, and to a lesser extent myoglobin, but it's a good contrast to hemoglobin, is it's the classic case for allosteric regulation, or allostery. Both of these bind oxygen and facilitate the dispersion of oxygen to our tissues. The big difference is that hemoglobin is a oxygen transport protein, and myoglobin can more accurately be thought of as an oxygen storage protein because it has a higher affinity and doesn't let go of the oxygen as readily. So if we look at these binding curves just below, where we're looking at myoglobin in blue and hemoglobin in purple, you can see a very, very different kinetic for the way it's interacting with the oxygen. And myoglobin has this classic curve to it that you would see for non-allosterically regulated systems, and then for hemoglobin we have this sigmoidal curve. And it's really it's important that we focus on the biological function of the hemoglobin in this case, because this sigmoidal cooperative binding of oxygen by hemoglobin is what allows it to function biologically. It's important that at the lung, where we have a higher concentration of oxygen, that the hemoglobin can grab that oxygen, but then that it holds on to it as it passes through the body until it gets to the capillaries, where it has a lower tissue density of oxygen where it can start to release it. I also want to highlight quickly the structural differences between myoglobin and hemoglobin. Whereas myoglobin is just monomeric and relatively simple structurally by comparison, although it contains a heme, the hemoglobin is tetrameric, which gives it a lot more ability to be tunable and also provides for its cooperative oxygen binding, which gives that sigmoidal curve. So what allows for oxygen to be bound by hemoglobin is heme groups. So heme group is an iron-containing, highly conjugated system, which we see over here on the right side. And that iron is coordinated to histidine residue, which gives it, the iron's location, a unique ability to influence the orientation of these residues above and below it. So this is considered the heme plane. So we've got the plane we're looking at on the top on the right side, and then if I were to tilt it down away from plane, that's what we're looking at here. So myoglobin, being thought of as an oxygen storage protein, has a much higher affinity for oxygen at all levels of tissue oxygen pressure. It's going to hold on to that oxygen until it gets to a very low oxygen environment, and then it will release it, and only then. So it's going to hold on to it and store it. Hemoglobin wants to bind the oxygen in the lungs and release it strategically under normal physiologically low conditions in the capillaries and the peripheral systems. So hemoglobin becomes saturated with oxygen at the lungs, and then at a place where the partial pressure of oxygen is about 100 torr, and then at around 40 torr when it hits the capillaries is where it's released. And it can do this because the binding of the oxygen to the heme by hemoglobin is cooperative. It binds the first oxygen to the first subunit of the tetramer. This makes the next one more favorable, and makes the next one more favorable, and makes the next one more favorable. And then it wants to hold those until it gets into that 40 torr zone, and then one falls off. And then as soon as the first one falls off, it makes losing the second one easier, which makes losing the third one easier, which makes losing the last one even easier. So it's this cooperativity and binding and releasing which really changes that, gives us that sigmoidal curve. And this is just a diagram showing some of these little conformational changes. I guess they're not that little. 15 degrees of rotation is pretty significant between deoxymyoglobin and, oh, these are myoglobins, deoxymyoglobin and oxy. No, sorry, this is terrible eyes. Deoxyhemoglobin, good, and oxyhemoglobin, so me. This is our deoxy version in A, and then once it's bound to oxygen completely, you see these rotations to its structure. So when oxygen is not bound to the heme, the iron is actually up out of plane a little bit. And we're going to take a look at that on the next slide. And when it binds oxygen, it pulls the heme down into plane. And when it does this, it pulls some of the ligands associated with it. And you wouldn't think this is such a big deal, but if you pull this residue with the iron, what it does is it causes the change to a whole bunch of salt bridges within the protein, and very significantly changes its chemistry. So here's what we're looking at. If we look at the binding of oxygen down here, without oxygen bound, we have this bent up out of plane state to the heme group because the iron is up out of plane and therefore pushing this histidine F8 up out of plane as well. When it binds the oxygen, the iron gets pulled down into plane, and this pulls the histidine residue with it, which changes the orientation of the helix, which has the histidine bound to it. And over on the side here with A and B, we see a couple different salt bridges diagrammed that all are modified by this oxygen binding event. This is just a quick resummary of things I've been saying, so you're welcome to peruse, but it's not worth necessarily spending time saying again. Now, some of the things that regulate hemoglobin, we've been talking about its function and the way it works, and I mentioned it's really important for allosteric regulation. But what is allosterically regulating hemoglobin? Well, one of the most important factors is the presence of free protons, which is also the acidity of solution. So, if you look at this chart over on the right, you can see, compared to myoglobin under all conditions, hemoglobin lets go of its oxygen at much lower percent saturations oxygen. But if we look at this pH dependence, 6.8, 7, 7.2, 7.4, 7.6, you can see that as the concentration of protons increases, because remember, pH equals negative log concentration of protons, meaning a low pH has a high concentration of protons, or higher. Here we have more protons, here we have less protons. You can see that the hemoglobin is giving up its oxygen more readily at higher concentrations of protons and holding on to the oxygen more readily at a lower concentration of protons. And this plays into the fact that there's oxygen binding is affected by a lot of different agents, not just protons. And so what we're going to discuss, we'll talk about protons, we'll talk about CO2. We won't pay too much attention to the chloride necessarily right now. But the reason this happens is because deoxyhemoglobin has a higher affinity for protons than the oxygenated version of hemoglobin. So as pH decreases, the dissociation of oxygen from hemoglobin is increased. And this is a little bit of a simplification because it's not good for stoichiometry, but you can see here that we have this equilibrium between the oxyhemoglobin and proton and the deoxyhemoglobin and CO2. So CO2 also promotes the dissociation of oxygen from hemoglobin. The hydration of CO2 in tissues, let me see here. Hop down here. This is nice. Just going to hop around on you guys a little bit. Let's tie this all together and then we'll come back to this again. So when you dissolve CO2 in water, you get a reaction forming carbonic acid, which automatically dissociates to bicarbonate and proton in water. So this is the whole generalizable effect. This is enzyme mediated, but it's also spontaneous in nature. When you increase the degree of CO2 dissolved in water, you're going to increase the acidity of that system. Therefore, you'll have increased concentrations of protons as you dissolve CO2 in water. This is what ties CO2 concentration to acidity and ties both of them to the consumption of oxygen, because if we need to release oxygen, it's like we have high levels of CO2, that means we need more oxygen physiologically to balance it out. And so a high level of CO2, and therefore increased level of protons, both would indicate to hemoglobin that it's time to release oxygen. So going back up, you can see here in these curves that hemoglobin is more readily hemoglobin is modified by CO2 in its function versus lack of CO2. And we also have another factor here called BPG, which we'll get to in a little bit. BPG is bisphosphoglycerate, and it deserves its own attention, which we'll get to in a little bit. But this curve is just showing the change of hemoglobin plus CO2. And then we have hemoglobin in the absence of CO2, or BPG. And you can see that these lower curves are releasing oxygen much more readily than the non-CO2 bound hemoglobin. Already gone through this, which was nice. So let's talk about BPG, or 2,3-bisphosphoglycerate. So this is a really important allosteric regulator because the orientation of 2,3-BPG actually changes the entire kinetics of the allosteric regulation of hemoglobin. And if you take BPG out of the picture, you actually get this odd rectangular hyperbola structure to the kinetics. But that's just not the case. We always have a little bit of BPG going on, which is because BPG is actually a product of glycolysis. And so it has an incredibly important role in terms of our primary energetics, but it also has an incredibly important role in terms of our oxygenation and distribution of oxygen. So what's really slick about BPG is it actually integrates our respiratory system with our energetics. It's a really, really interesting biological feature. And what happens is BPG actually binds. So BPG has two pair of phosphates tied to a glycerate and is highly negatively charged. And it just plugs itself right on in the middle of the system and coordinates to a whole bunch of positive charges and really refines the structure of the hemoglobin. So that's really all of the slides that I think are relevant on this content. This is going to have been, I guess it's 52 minutes, and we hopped around a little bit doing it, but what I want everyone to refocus on, going back to the beginning, are these initial questions. What types of regulation are there? Focus a lot on covalent, specifically phosphorylation events. Focus on allosteric events. Focus on the two models that describe allosteric events. And then we have our two systems that we discussed. We talked about glycogen phosphorylase as a good example of a common enzymatic system, which is both allosterically and covalently modified and regulated. And then we talked about hemoglobin and how it's a classic case of allosteric regulation and cooperativity. So go ahead and just dive into this. And this is it for enzymes. The next two lectures are going to be a short lecture on carbohydrates and a fairly short lecture on lipids and membranes, which you're always balancing things, but everyone should be in a really good shape following this content for biochemistry too, which is metabolism, which is going to tie all this together for everyone. So we spend a lot of time in enzymes, but it's really, really important that we've got it because it's going to be leaned on heavily next semester. And any discussion of metabolism is going to need a lot of discussion about enzymes and their function and their regulation. So I hope you guys liked this content. It's some of my favorite in the course, and I'll be talking at you about carbohydrates in the near future.